Hello, everyone, and welcome to Lunch Break Science. I'm Ariel Johnson with the Leakey Foundation, a nonprofit dedicated to supporting human evolution research and sharing discoveries in programs like this. We'd like to thank the Ann and Gordon Getty Foundation and Camilla and George Smith, whose generous support made this episode of Lunch Break Science possible. Here with us today is Leakey Foundation grant recipient Deming Yang. Deming, thank you so much for being here with us.
Glad to be here.
Deming is joining us from Utah, where he is a postdoctoral researcher in the Department of Geology and Geophysics at the University of Utah. He uses stable light isotopes to examine changes in diet, habitat, and body water of fossil mammals. And this helps him reconstruct what past environments look like. And also, it helps figure out how changes in the environment may have impacted human evolution. This research takes him to the Kubifora Formation and east of Lake Turkana in northern Kenya, where he's conducted fieldwork at several sites. So really cool fossil he's holding there. He's also conducted research at the National Museums of Kenya in Nairobi, where he's worked with their extensive collections, which is super cool. I look forward to talking more about that. Today, he'll be discussing stable light isotopes and the kind of questions that can be asked by them, or answered by them, not asked by them. So before we hear from Deming, if you are watching us live on YouTube, Facebook, Leakey Foundation Live, or Twitter, you can ask questions or make comments for Deming anytime in the chat, and he'll be answering those questions live during the episode. The earlier you get those questions in, the more likely he'll be able to answer them. So be sure to get those questions in early. So my first question for you is, you use stable light isotopes to understand, the changes that are going on in the environment using fossil mammals. But what does this have to do with human evolution?
So, human evolution doesn't really happen on its own. I feel like this is something that, we as paleoanthropologists care a lot about. So, the fossil mammals sort of appear on the landscape as early humans did. And they also sort of tell us information about what kind of environment that they live in. And, and when they're associated with the human fossils, then we can say something about, what the environment was like. So, for example, we have species on the landscape that are more grassland adaptive, such as the zebras and the wildebeests. And we also have species that are more forest adaptive, such as the monkeys. And so once we see those different mammal species in the fossil record, then we can generally say something about what the environment was like when humans showed up, for example. And a lot of it has something to do with the general change in the landscape. So during human evolution, which is over multiple millions years of time, there was a recognizable climate change where the climate was getting drier over time, and so a lot more grassland is present compared to earlier times. And so such change in the landscape is also preserved in the fossil record with the fossil mammals.
You split your time between the field and the lab. What gets accomplished in both of those locations?
So in the field, we generally do a few things. So to put it in a very simple way, first of all, we go out to the field, go out to the outcrops. We look for fossils, and here's me holding some fossils in my hand. And once we locate some fossils, we will try to understand where they come from, because the fossils themselves only tell us, you know, a limited amount of information, but we also need information from, say, where the, you know, the levels in the stratigraphic sequence where they come from, so that we understand how old they are and their relationship to other fossils we find at the site. So once we have all this information, we will collect the fossils and we'll bring them to the museum. So, but this is just to put in a very simple way. And as you can
Mm.
imagine, you know, like any kind of a road trip, things can go south very easily.
Oh, I
So
bet.
yeah, so here's some photos of my field work where we forgot to bring a kitchen knife.
Ooh.
So then we have to improvise for a bit and try to cut our vegetables with machetes. So well, that, you know, was an quite interesting experience. Yeah, for sure. And in the field, we also often work with students. You know, for example, we may have few school students coming along and they'll be learning from us about field techniques and what's going on in there and have like a firsthand experience for themselves.
And then
Um,
yeah, I was gonna say,
yeah,
is
so
that when it goes heads to the lab?
yeah, so like once we have a lot of the stuff that we collected from the field, and the first thing that we do is to bring them to the museum. And at the museum, there are a lot of things we can do to the fossil specimens. And for example, we can measure the sizes and, you know, take notes on the different fossils that we have found. And also, we can carry out some sort of analyses that we can do on the fossils. For example, for myself, for the work that I do, I play a little bit of a paleo dentist. So by doing that, I have like a little dental drill and I scrape the surface of a tooth so that I get a little sample from the enamel. And with that sample of the enamel, I go to the lab, and in the lab analysis, then we can figure out the chemical composition of the tooth that tell us something about the stage of life, the general stage of life of that animal when the tooth was forming. So so yeah, the whole process in the lab takes quite a bit of time. It's quite a bit of an effort because, you know, you have to wash the sample. Sometimes dust can get in. You know, you don't want to eat the dust for,
Mm.
you know, the machines won't be happy about that. And then, like, once we have the numbers from the machines, then we can say something about, you know, the animal that we just sampled the tooth from. So, yeah, so that's sort of the general flow of work that we do.
How do you determine which teeth to use and how many teeth do you need to analyze?
So, so when we're in the field, teeth can show up in various different shapes and sizes. And the first sort of piece of information we can get from them is what type of animal the teeth are coming from. And so, for example, we talk about the different species they're adapted to forest or grassland kind of environment, and this is sort of the first step. So we identify them. We, you know, have a good idea about like what animal those teeth are representing. And then once we have that information, once we know what the animals are, then we can take a very educated guess about like what, you know, environment they're adapted to. And in the case of the analysis that I was talking about, taking tooth samples or enamel samples, is that we can just guess what the diet is for this particular animal. And in the, you know, in the diet sort of part of the story, we can generally say whether they are forest adapted or you know, grassland adapted. By saying that, okay, this animal is probably eating a lot of leaves versus grasses. So For the sampling, there are a lot of ways you can sample the teeth, but in general, we need preferably a good number of them so that
Yeah.
there are multiple samples representing one particular group of animal that we're interested in. And I would say we need at least 10 individuals so that we can have like a good idea or a good coverage of, the understanding of the diet that, of that animal.
Do you ever
So,
yeah, do you ever run into the problem where you don't have enough teeth to analyze?
yeah, so sometimes it happens because, it happens when there are not a lot of animals available, like not a lot of teeth available from either a fossil collection or from the field. So then, sometimes we have, a lot less teeth being represented. So if that's the case, then, we just have to admit that because the sample size is pretty small, then we are less confident about
Mhm.
what we can say about their diet or their environment, things like that. So we just have to be very honest because, this is how science works. And yeah, and perhaps we can also suggest that future work is
Mm.
going to bring more samples to our knowledge, and that is going to help move the science along.
So we've heard a little bit about the background of your research, but I'm really curious about your background. When did you know you were going to be a scientist?
Um, so I've always been interested in being a scientist. I would say, like, I was fairly sort of curious about the world around me when I was growing up as a kid. I like went to the pond and caught some like tadpoles and fish and whatever and studied them. A lot of it was like butchering them in a way. As a little kid, like there's not really much I was appreciating about life in different forms and and by destructive nature of some kids like myself when I was a kid. That's what I did, and but that was in general like really satisfying a lot of my interest in the world and especially in life sciences. This is what I um, got to pursue as an undergrad. So I was really interested in different organisms and how different organisms work, and like from their development to their evolution. There are just like a lot of things out there I was fairly interested in. So and um, from there, I also learned that there was probably quite a bit of lab work involved. So, in a sense that, because a lot of it was about being in the lab, doing analyses, and, culturing different material like cells and bacteria. A lot of times we use those as our tools,
Yeah.
and that lab work seemed to me like, or that sort of lifestyle or schedule, research schedule, seems to be departing from my very genuine and childhood kind of interest that I had. So in a way that I always wanted to go to the field
Yeah.
and be with nature a lot. So I thought that maybe it's time for a little change, and so yeah, that's why I chose to study paleoanthropology.
So what was your first experience in the field?
So in terms of field experience in paleoanthropology, my first experience was in with the Kupi Flora Field School in 2011. So it was a pretty inspiring experience in many ways. And, we just went to the field with like more than 10 vehicles and, students and instructors all together. Um, and, I got to learn about what's going on in the field, some of the methods. And I think the part that I was really really happy about was the opportunity to interact with the instructors at the time, and they were very nice and very knowledgeable. So I felt that I learned a lot from them. And so at the beginning of the field school, we were going to areas of Kenya where you can see like the sort of the modern landscape that we have today, like the savannah kind of a landscape, and with animals. So we see the animals, we understand like what they're doing, and then we also look at some of the animal bones that are being left, say, just on grassland somewhere, right? Like, so their bones popping up because some animals died, say, a couple of years ago or something. And we would basically go look for some animal remains, and we're trying to understand how the fresh bones could become fossils, and in like what kind of an environment it could be. So that was fairly interesting, and then there was good exposure to what bones look like as well from the field. And another experience I really like about the field school was, there was like this little workshop thing that we did, and we were just going over different animal bones at the Kubifora camp. And, there are a lot of them, and
Oh,
we
yeah.
have to learn how to identify them. And so I'm still pretty proud that I did fairly well in that activity. And I thought that was like a reinforcing element for me to, continue my pursuit of paleontology or paleoanthropology as a career.
Yeah, fossil identification's got to be pretty difficult.
Um, so I guess sometimes identifying bones and teeth could seem a little daunting to people, but like I feel like we could give it a little try here as like a an activity, an interactive activity between us and the audience, and see how we do. Yeah.
Yeah, let's do it. So we have a fossil from Kubifora that Deming will be sharing with us. We'd like all of you viewers watching live to guess what this object might be, and by submitting your guess in the chat. So, Deming, what might help our guests or our viewers guess what this is?
So, a lot of it, so when we're picking up sort of any sort of an object from the field, the first few things that, we want to consider are for, the shape
Mm,
of the object. And this is where a lot of information can come from. So, for example, does it look like bone? Does it look like, a tooth somewhere from some sort of, animal out there? Is there some other sort of interesting shape that we can recognize from this object? So the shape by itself is a great source of information already, and a lot of the times, shape itself is enough for identifying an object from the field. And there are other sort of cues that we could use, like color. So sometimes objects can show up in colors that you might have seen before, like
yeah.
bones tend to be a white color, but fossils might be different because the fossilization process would stain the bones in a way that the color is going to change. And there's also texture. So like, once you touch it, let's say touch is a,
Oh, yeah.
sensation that is very important in terms of getting the information from the object. So it really helps to be able to hold in your hand, for example,
Yeah.
when you are trying to identify an object from the field.
Well, we have some guesses. Let's get see what people have assessed this object to be. We have a gastropoda.
Hmm.
Let's see here. What else do we have? Shell,
Hmm.
snail shell. Seems like we have a theme going here.
Yeah.
Another. Okay, so it seems like the vast majority of our assessments are that it is a gastropod shell. So
Hmm.
what is the answer?
That is correct. So congratulations for those who responded, and this is uh, without any doubt some sort of fossil shell from the Kupi-Flora Formation. But the interesting thing about fossil shells in the context of sort of in the outcrops is that it tells you what the environment was like
Yeah.
when the shells were buried. So it tells you that the environment was probably some sort of freshwater environment, like a lake or a river kind of a situation when the shells are being buried. And it is really the tectonic activities of the site that brought the sediments on the surface again because they would have been buried deep. So then with erosion and other processes going on, then we see shells and other fossils on the surface. So yeah, the shells definitely tell their own story. They're not just some sort of random things that we see in the field.
So now let's go on to round two, which oh, may be a little trickier. What might help our
Yeah, this
viewers?
one this one is pretty interesting. So I mentioned something like the shape of the object, the color, the texture.
Mm.
So the color isn't really that interesting. It's you know pretty much the same color across. There's a little bit of a texture here. You can see some roughness on the surface, but you can also see some distinct patterns that kind of look like scars on
Ooh,
the surface. And the shape is like an oval shape. But something that I want to say here is look at its size. Its size is fairly interesting because it seems to sit pretty well in the hand, right?
yeah, it definitely does.
Yeah. So yeah, these are the things that may offer some sort of a clue to identifying this object. And do we have some answers coming up?
We're still waiting for some answers, so get your answers in. I know this is a tricky one, but
Yeah. Yeah, I was just looking at it again and trying to think like what other features that may help with the identification.
okay. Well, we have a couple of we I now have a couple of answers that have come in. People have become less shy. Okay, let's see. What do we have here? We have stone tool. We
Okay.
have a stone tool.
Mm-hmm.
We have let's see what else do we have? Another stone tool
Mm-hmm.
and lithic scraper. So what is our answer?
It is a stone tool. So great job, everybody. And one of our viewers also mentioned that it's a scraper, and
Yeah.
this is the correct answer. First of all, it is a scraper. And the features that sort of go into the identification are actually around the edges. So there's this one surface that is facing us for the stone tool, that is relatively flat, but around the edges you can see multiple scars where you know and because it's a stone tool, it was worked by an early hominin. So around the edges, you can see that there are the remnants of the hominin's work in a way that flakes were being knocked off from the edges. So either this tool was being used as a stone core to produce the flakes, or this core was the tool itself. As you know, as I said, it fits very nicely in the hand. You can, use it to scrape the hide of an animal or something using the rough edges that the hominids created. So that was a really cool. So like a piece of stone tool by itself can tell you a lot of stories about like its making and like
Yeah.
who the maker could be, for example.
Well, let's go on to our round 3. This one looks,
Yeah, this is an interesting one.
yeah, very different than the first 2 rounds. I think this has definitely gotten harder as we've gone along. So what about this one would help our viewers?
So again, we are looking at the shape, the color, the texture of this object. And in terms of shape, I would say this is definitely not looking like a bone. So it doesn't really. So for those who are familiar with like chicken bones or your dog's toy or something, this doesn't really look anywhere close to it, first of all. And the color, there's not really much to say about it. It's fairly pale in color from the outside, but the color can change over time as sort of like the weathering process is going on with fossils and other objects in the field alike. The texture is interesting. It's kind of so towards the right side of the screen, it's a little tapered, and it's also the shape of it again is a little like rounded and cylindrical, I would say. So yeah, this is an interesting one. But something I can actually say for sure, or at least to offer a hint to our viewers, is that it's not a bow and it's not a tooth. So that leave us with some other options out there. And yeah, so do we have some answers?
Yeah, we do.
coming up?
I think we've got some paleoanthropologists out there. So we have two guesses, and they are both coprolite. Oh, and there's been a additional onto the coprolite, a carnivore coprolite. So yeah, seeing that now that we've said the answer, people are adding answers. So no, okay. So what is the correct answer?
Everyone got it correct. It's a coprolite. So a coprolite is fossilized poop, and I like one of the viewers specifying that it is a carnivore coprolite because carnivore poop is more often uh, better preserved than other sorts of poop out there because, like, say, if you imagine, if you encounter some sort of hyenas and jackals, in the Serengeti, and their diet is mostly animal tissue, first of all, and also they eat a lot of bones. So, because their diet is, already very sort of like dense in a way, high in minerals. So their poop also tend to be fairly dense and high in minerals, and in that sense, their poop gets preserved much more frequently than the herbivores out there, like the wildebeest and the antelopes. So yeah, a lot of the times when we see a coprolite, um, from the fossil record, it's most likely going to be that of a carnivore.
Well, this was a fun activity, but I'd like to get back to hearing about your background. You had an amazing opportunity to live in Nairobi for 3 years. Could you, yeah, what was it like?
So to be in Nairobi is like any sort of study abroad kind of experience. First of all, you are being immersed in a very different kind of a culture. Maybe, you know, there are the people who you would interact with are, completely different from the people that you know growing
Yeah.
up. So first of all, it's like an experience to adapt to a new environment. And also it offers a lot of opportunity to learn. For example, you learn about, the local culture, you learn about their language, you learn like, the things that they care about,
Mm.
the things that make them happy, make them sad, like just be a person and be a person with your friends around you. So that by itself is completely different from any sort of experience growing up. And so for a lot of the time when I was living in Nairobi, I was working at the National Museum Kenya. So you know that experience, because I was participating in a lot of museum work, working with the fossil collection there. There's a lot of opportunity to learn from the fossils and also from visiting scholars. So it was a really nice experience because the Nairobi Museum is by far very often traveled by paleoanthropologists because of just how extensive the collection is. So I had a lot of experience interacting with visiting scholars, um, learning from them. So that was very good. The other part of being in a museum is that fossils are not everything. So, although I spend most of my time with the fossil collection at the museum, I also had some opportunity to interact with people from other sections of the museum. For example, from the people from the ornithology section, the mammalogy section, and the osteology section is probably the most relevant because a lot of us study bones, and bones are everywhere,
Yeah.
and people have collected them, so they are stored in the osteology section. So we visit there quite often, actually. And that sort of points to the nature of paleoanthropology being a multidisciplinary field, and we have to learn from like everyone basically. So yeah, so that was a good opportunity for me to learn. And yeah, living in Nairobi also offered a lot of sort of opportunity to go out of work and to because being in a different country and being in a different environment, I was really enjoying a lot of work with Nature Kenya, for example. The photos are showing my trips with Nature Kenya, and I was fairly interested in photography. So we went out and looked at birds with Nature Kenya, and there's been a long-term program going on with Nature Kenya, doing some sort of water bird surveys around big lakes along the Rift Valley.
Oh, it's such a
So I
cool
was
photo.
I was very fortunate to be able to go with them and learn from them about like bird identifications and their migration patterns, and also like these are just really fun activities. And out of work, like because you can only be in the museum for like 5 days a week, right?
Yeah.
So yeah, that's what I did outside of work. And also around Nairobi, there are a lot of things like, Nairobi National Park, places that you can visit. I remember there was this one time, I was at the Safari Walk somewhere close to the Nairobi National Park, and it was lunchtime for
Ooh.
the lions, and they were having a lunch break, and I was having a lunch break science episode with them. So in the photo that we just saw, I was holding like a big chunk of meat, and it was because the workers there were just going to feed the lions. I was like, well, may as well try to understand how much the lions are eating. So I was trying really hard to pull that piece of meat really close to my head.
Yeah, it looks
And
heavy.
that shows you that how heavy that piece of meat is. And there is some other things in the bucket over there. So yeah, the lions have a big appetite. Um, so the other experience I really liked was going to Mount Kenya, and just because Mount Kenya is such a cool place, it's like a little island of an ecosystem in the middle of like um, what we know about Eastern Africa today as like the savanna kind of a landscape. So it's very different from there, and a lot of very special plants and animals that I saw up there. A lot of endemic birds. So yeah, there was all of these things were like very enjoyable, very fulfilling, and yeah, I absolutely enjoyed that.
Well, it must have been just an amazing opportunity. When did you become interested in stable light isotopes?
So yeah, so as I mentioned, at the museum there are a lot of visiting scholars. So I was fairly inspired by Professor Tori Serling at the University of Utah, and I had a little overlap with him when he visited Nairobi, and you know it was a very interesting experience because he was sitting in the hominin vault, where the fossils are. It's more like a lot of scholars go, and it's like a sacred, almost like a sacred place where a lot of the early human fossils were
Oh,
stored, for the researchers to come and study them. So he was sitting in the room and holding a drill in his hand, like a little handheld drill, uh, you know, and I was just out of curiosity asking him what was that about, and then he told me that, oh, I was just playing a little bit of paleo dentist. So that conversation that I had with him really sparked my interest into stable light isotopes and what they can do to inform us about like the diet of animals or the environment. So yeah, I thought that was the sort of my biggest inspiration, and also where like my research interests, especially the things that I do today, my research where it began.
what exactly are stable light isotopes?
So stable light isotopes, it's quite a bit of a mouthful.
Yeah.
So first of all, they are isotopes, and isotopes are atoms of the same element, but they are of different atomic masses. So in this example here, we have an element hydrogen, and there are isotopes as hydrogen 1, hydrogen 2, and hydrogen 3. So again, they're of the same element, but they are at different atomic masses. And so these are, isotopes that we are mostly dealing with. And so apart from the word isotopes, we also have stable. Stable meaning that it is not radioactive, because people are probably most familiar with radioactive isotopes, like what you have in you know, atomic bombs or nuclear plants and places like that. So they are radioactive, uh, being like not stable, meaning that they will generate heat or energy as they decay. So the decay process basically means that they are not stable. So what we do is we study the opposite of that. We study the stable ones. They will not decay. They will not change into a different element as time goes. So that's what we study. And the other word in there is light. So the light is just describing what the elements are. So the most common elements that we have for studying stable isotopes are hydrogen, carbon, nitrogen, oxygen, and sulfur. And these are, for those who are familiar with the periodic table, those are colored in blue. So the reason why we study those light elements is that they are the building blocks of all living organisms. So if we study anything that is made, for example, that is a living organism or that used to be a living organism, then they will have to have those elements in their body. So it's like we are looking at what is left behind by uh, either living organisms or organisms that are once living, and though those evidence can then give us some idea about what is going on in the organism.
How accessible is stable isotope analysis?
It's we have seen a lot of growth in the field of stable isotopes, and in terms of their applications, they have been applied to multiple different fields such as paleontology, archaeology, ecology, and even forensic science. So one of the projects in my research group right now is trying to use stable isotopes to help identify service members that fought like wars outside the United States. And it's really based on a very similar principle that we talk about. Teeth preserve a lot of the the general state of life for uh, the individual when the tooth were forming. So using some remains from the service members, um, we can help to identify where they could have come from, or then we can contact, perhaps once we narrow down to where they come from, we can contact their families and help to return the remains, for example. So a lot of it is very meaningful, and then I feel
Mm.
like the applications is of stable isotopes really help us understand a lot of different things, and not just things about science, but also things about people and about their stories. So yeah, we have seen a lot of growth in there, and it is becoming more and more accessible.
Well, now let's turn the virtual floor over to you, Deming, and hear more about stable light isotopes.
Thank you. Okay. Um, Let me see. All right. So the topic of our discussion today is how to be a detective with stable isotopes. So we know that, as I mentioned before, the elements that are the main building blocks of all organisms are hydrogen, carbon, nitrogen, oxygen, and sulfur. And as organisms come and go in Earth history, they always stay within the very large and complex system called the biosphere. And as energy and elements flow through the biosphere, they tend to leave behind the trail of breadcrumbs for us detectives to figure out like what exactly is going on in different steps of the system. So this is the idea of being a detective using stable isotopes. Stable isotopes have um, the characters of a tracer, for example. So let's start with something that we are all very excited about: food. And who doesn't love food, right? But I feel like perhaps the most profound question we can ask ourselves is what to have for dinner. So uh, so let's take a look at what we have here as food. And on the left side of the screen, we have some nice looking vegetables and really beautiful fruits, and a lot of the grains that we love dearly. Very high starch rich grains out there, rice and beans and stuff. And on the right side of the screen, we have something that we are also very familiar with. We have sugar cane or cane sugar and corn. So the reason why I isolate these two elements, on the screen is that when we measure the different foods with stable isotopes, in this case, it's the carbon isotopes because all of these foods will provide us with energy in the form of sugars. Then we will see a pattern showing up that the sugar cane or cane sugar and corn are fairly different from the rest of the food pile. And what we do is we take a magic measuring tape and we can generally tell where those food items fall within the spectrum of the measuring tape. So, for example, most of the foods we're familiar with fall on the left side of the spectrum, and corn and sugar fall on the right side of the spectrum. So now that we know that there is a difference in sugar and corn, like how do we use this information um, uh, as detectives? How do we, figure out the story behind things that we care about? So here I give an example of whether there is added sugar that we can detect in sparkling wine. And I'm sure everyone loves sparkling wine. And we will use the same sort of magic measuring tape here, and the cube, sort of like the squares that you're seeing on the screen, there are some white squares and some black squares. They represent individual samples of sparkling wine. And if we already know, for example, that grape juice fall on the left side of the spectrum, and cane sugar generally fall on the right side of the spectrum, then we can pretty much just tell which samples or which sparkling wines have added sugar. In this case, that would be the ones represented by the black squares. And guess what? A lot of those sparkling wine are coming from Australia, Brazil, and the United States. So, this is a really good tool for a detective in a way that we can figure out what's actually going on in our food. So, how do we apply the same kind of principles to the paleo landscape when we're studying paleo environment? So, it pretty much works in the same way as we discussed in our food story. So in the tropical environments of Africa, different plants also fall along the same spectrum, but at different places. So, for example, we have trees falling on the left side of the spectrum, and we have tropical grasses falling on the right side of the spectrum. And with this information, we can then try to sort of figure out what the animals are on the landscape are associated with, which uh, environment, for example. Is it forested environment or is it grassy kind of an environment? And we do that by measuring their teeth. So if we measure the teeth of, say, of a giraffe, then it's showing that the giraffes is eating a lot of the leaves. And if we measure the teeth from zebras and wildebeest, then they will show up as like, pretty much more like a grass when it comes to, where they fall on the spectrum there. So the idea is like you are what you eat. So as the giraffe is eating leaves, it's going to sort of inherit the pattern that we are seeing on the magical tape measure there, and it's going to show like what the diet of this animal is. So pretty much the same thing we can tell of the hominins, because you know they also follow the same kind of principle, and they live in habitats that are somewhere in between a leafy forest and a pretty open grassland. Okay, so now that we know the story about food, how about water? And then, as we know, water is arguably the most important resource for any organism on Earth. And so, what are the questions we can ask about water as stable isotope detectives? We can use the same concept of a metrical measuring tape. For example, we know that ice on land for on the left side of the spectrum there, and water in the ocean for on the right side of the spectrum. And if we were to put all of the water together from the world into one sample, in a sense to average everything out, we know that there is a lot of water in the ocean. So the Earth, as like, average of all samples out there, is probably somewhere really close to the ocean. So in the sense that, along the spectrum there, the Earth will fall really close to that of the ocean. So you can think about this as like a system, the balance between ice on land and water in the ocean, as like a system similar to a lever with the Earth in the middle serving as the fulcrum. So, what does it really mean for us, and what kind of a story we can tell based on this sort of a balance here? And now enter the Ice Age, where a lot of the North America is covered in ice sheet, and also when mammoth, sloth, and saber-toothed cats go on a road trip. So during the Ice Age, we know that there is a lot of ice on land. So you can imagine we are piling up a lot of ice on the left side of the spectrum. And this will tend to sort of tip the balance towards the left side. But we know that the amount of water, the total amount of water on Earth, is not going to change, because water is not really going anywhere. It stays in either one form or the other. So as there's more ice piling up on the left side, we also have a smaller amount of ocean water, because we know that the sea level was a little bit lower during the Ice Age. So what it means is that during the Ice Age, the oxygen isotopes in the water will have to move towards the right for the whole system to stay balanced. So this movement is a good sort of indicator of how much ice there is on land. So we can use this information. For example, if we see that there is fluctuation in the oxygen isotope composition of the ocean water in Earth history, then we know that there is some fluctuation in global temperature. So in the sense that this will serve as a paleothermometer if you're interested in, climate change and things like that in Earth history. Okay, so that's the end of discussion today, and I hope everyone enjoyed it. And I just want to say that this is meant to be a very brief introduction to the tools that we have as, detectives or isotope detectives, and looking for evidence that we could use to reconstruct environment of the past. So there are, definitely a lot more details in the research where similar principles can be applied. So the sort of uh, the measuring tape sort of a system that I talked about. So if you're interested in learning more about this, stay tuned for more episodes of Lunch Break Science, where my colleagues will continue to talk about their research. Thank you.
Thank you. That was a really fascinating kind of introduction, and like I now I'm like I feel like I want to go run to my see what my sparkling wine, where it's from, and where the isotopes fall on that the meter. We'll be taking some questions from the audience, so if you have questions, please be sure to get them into the chat now. I have a quick first question though. So you are working on more modeling and method development right now. Could you tell us a little bit about that?
So, yeah, the modeling work I started very recently because, a lot of it has something to do with not being able to travel because of the pandemic. And I thought that, modeling, first of all, doesn't really require a lot of new data because we already know a fair amount about what's been going on in a lot of the sort of systems that we're interested in. So an example on the screen there is just some plant physiology. So plants will respond to multiple elements in the environment. And the modeling work is just to model each every step, what goes in and then what comes out. So and we study that in an isotope sort of a fashion. So it's similar to like when you have multiple knobs as like a DJ somewhere and you turn them and it's gonna you know uh, do different sort of effects to the end result as like the mixing of different soundtracks, for example. So we are doing something very similar to that. We are turning the knobs. Since we know some of the systems fairly well, we can sort of try to identify what are the elements that matter the most to the variation we see towards the end of when we're taking the measurements of the system, for example. So again, like I said, this is a good practice for understanding what matters to the system, first of all. And also, I feel like it's really worth noting that it's very important for us researchers who go to the field to sort of be respective of the countries that we work in, because we know that the United States has a fairly high vaccination rate, but we don't want to be the people who go travel and spread a virus somehow. This is just, a very bad practice, at least. So yeah, the reason why I took on the modeling work is that I can avoid as much travel as I could because, we already have a lot of data just, by the decades of research before us. And by doing that, we can also understand the system better.
Okay, well, now we will take some questions from the audience. If you have questions, get those questions into the chat right now. Do we have we have a first question from Valerie. She asks, if a tooth is fossilized, how do you get information from it as far as diet, etc.?
That is a really good question. So first of all, I want to talk about like why we look at teeth, and the reason is that teeth are just everywhere. Teeth are probably the hardest tissue out there for mammals. For any of our body parts. If there's any reason any part of it gets fossilized, the most likely part is going to be teeth. So there are, it's just way more teeth out there than other bones that we have seen in the fossil record, first of all. And the other part of it is that when teeth are forming, teeth are fairly strong and mineralized because you use your teeth a lot and you use it for your lifetime, and they only wear down very slowly. So it's like in a sense, you probably have sharpened your kitchen knife multiple times, but you almost never really have to do a lot about your teeth because they just are so strong. So for a very sort of highly mineralized and strong sort of a tissue to be fossilized, a lot of the sort of chemical composition of the teeth are being preserved in the fossilization process, a little bit like different from other bones that we see because bones are very porous. There are a lot of holes in them, and minerals from the sediment can seep in and replace a lot of the chemical signature of the bones over time during fossilization. That's why generally we don't use bones in our analysis, but teeth are fairly good. They preserve fairly well, and they preserve the original dietary information in the organism when the teeth were forming.
We have another question from the audience. This is from Deepak. What is one single sample enough to tell whether paleofauna is C3 or C4, or we have to take multiple vertical samples from the same enamel?
That's a really good question. It touch bases on a few different topics that I didn't mention today. So first of all, what is C3 versus C4? So I was just trying to keep the language simple when I introduce most foods versus like sugar cane or cane sugar or corn. So the scientific term for those different groups of plants are C3 and C4. The same way it goes with the environment when we talk about forest adapted versus grassland adapted sort of a situation, like forest is mostly C3 and grassland it's mostly C4. So with that out of the way, then can we tell something, the diet of an individual when we take one sample from the tooth versus multiple samples? There are different strategies. So there's definitely more information we can get if we take more samples from a single tooth, because a tooth is being grown incrementally. You can think about it as like growing like a snail. Like the snail is going to grow kind of spirally, so towards the tip is the very early part of the snail, and like towards where the snail actually comes out, it's going to be like close to the life stage of the snail right now that we see. So teeth work in a very similar way. It's growing like in a sequential way, that when you take multiple samples from a tooth, then you essentially sample different parts of the life of this organism. So then, there's definitely more information we can say when we take multiple samples from a single tooth. But in a very general sense, we could also just tell like what kind of a diet this organism had by using one sample. So yeah, it's a matter of preference, and also what is the research question as well.
Well, I think we have time for one more question. This comes from Cheryl. Using technology you dream of having available someday, what one question about human origins would you like to answer regarding SLI?
What is SLI again?
We have a stable light isotopes.
Oh, okay.
Like it took me a minute.
Sorry,
I'm like SLI.
Yeah, I'm blanking out on that. Like sorry. Okay, good. So yeah, with stable light isotopes, I mean, it's actually not an easy question to answer. Um, by any means, because when we are working with stable light isotopes, we are working with some limitations. You know, there are limitations with stable light isotopes. For example, if we are looking at carbon isotopes, for a lot of the questions that we can actually answer, is going to be diet, right? Because it's about different plants and how animals incorporate different parts of the diet into their body and into their teeth. So the same goes with other isotope systems. So there are only a handful of information, I would say, that we can get from each. So then, it's totally worth exploring the possibility of using multiple of them so that we can have, say, one side of the story being told by one isotope system and another side of the story being told by another isotope system. So that's actually what a lot of people do. We try to interpret the data from multiple sources of isotopes, and that is going to give us a more like a complete picture, like say if you were trying to reconstruct the paleo environment. So with all of the sort of technological advances that I could have, I wish that we can just take an isotope measurement of any element that we want in the hand with the smartphone. One day we can just go around and zip zap, and then we get the numbers immediately. So that would be what I really wanted. But of course, this will it's still a long way, right? Like until we can eventually make something happen like that. So, but yeah, there are definitely a lot of things that can be advanced with science.
Absolutely. My last question for you before we close is do you have any advice for those who are watching who are interested in pursuing paleoanthropology as a career?
Yeah. So, um, as a career, I would say, yeah. So just speaking from my own experience, like we talk about me being in Nairobi for a while, and a lot of it I feel like has to do with trying to get out of your comfort zone, first of
Yeah.
all, because, I had some doubts when I was going into it. I was not sure whether it was safe, like I'm sure a lot of people had similar concerns when they go travel in different countries, just that, there's not very much information or a firsthand kind of experience from a personal perspective. So yeah, I thought that getting out of comfort zone and challenge myself with that was definitely the way to go. And then like thinking back, I have absolutely no regret. I learned so much. I got to know so many people, and I still have a lot of friends in Kenya. And whenever I go back, I really enjoy, being there because I was there. So yeah, a lot of it has to do with getting out of your comfort zone. And some other advice I feel like is, has something to do with, not just paleoanthropology, but in general, like a good life practice is to learn new skills. I learned a little bit Swahili by living in Nairobi, and I learned how to identify birds over there. So those things, I mean, they might not really give you a job, but like at least they are very fulfilling. I feel like I was actually learning something. It's very rewarding. I was able to identify them later. The birds. I enjoy watching them. So yeah, whenever there is an opportunity to learn, I would encourage everyone to just jump at it, like because we can only grow, intellectually, we can only grow by learning new things. So, and I guess the last part I want to say about this is try to be adaptive
Mhm.
because like I said, being in a different place, learning a new language is all about like how you want to fit like in the new environment. And then I feel like, this goes a long way from the very early parts of human evolution. Imagine our ancestors show up on the landscape and they didn't know anything, right? Like they probably had some things to work with, but quickly they will have to adapt and to survive. So the same goes with pursuing a career or you know um, just trying to be a person, right? Like
Yeah.
I feel like that's definitely part of it, and trying to be adaptive and make the most out of the situation, like the pandemic, for example. We're doing a lot of virtual meetings nowadays, which has a lot of drawbacks, but there are also advantages like, for example, we will be able to virtually invite speakers from, you know, all over the world, and they don't have to travel, but we can still get to enjoy their seminar, talking about their research. Maybe there's some other sort of good stories they can tell. So yeah, all of this is, be adaptive, persevere, and yeah, we'll get out of this. So.
Well, thank you so much. This was although I feel like I'm going to always remember you as the stable light isotope DJ. But thank you so much for being on the episode today.
Thank you. Pleasure to be here.
So next time on Lunch Break Science, we will be continuing to learn about the paleo environment with paleoanthropologist Enko Nagesh. It'll be a really great episode, and it'll be on May 20th. We also have a new episode of our podcast origin stories called Sleep in the Moon that will be coming out later today. So keep an eye out for that. That's going to be, I'm really looking forward to that episode myself about how sleep patterns are impacted by the moon. So thank you all for taking a break from your day and feeding your brain with the Leakey Foundation. Until next time, stay hungry for knowledge. Lunch Break Science is brought to you by the Leakey Foundation and is made possible by the generous support of the Anna Gordon Getty Foundation and Camilla and George Smith. Visit us at leakeyfoundation.org to learn more about the Leakey Foundation, today's guest scientist, and how you can help support human evolution research and educational programs like Lunch Break Science. Right now, all donations will be matched by generous donors, meaning your impact will be doubled. Miss an episode of Lunch Break Science? Catch up on past episodes and browse our library of Leakey Foundation lectures on our YouTube channel. Still hungry for science and can't wait till our next episode? Check out the Leakey Foundation's award-winning podcast, Origin Stories, available wherever you listen to podcasts. Subscribe to the Leakey Foundation's YouTube channel, follow us on Facebook and Twitter, or sign up for our newsletter to be the first to hear about exciting upcoming episodes and programs, as well as groundbreaking discoveries in human evolution research.